The Gabriella Miller Kids First Pediatric Research Program is an NIH Common Fund program whose goal is to help researchers uncover new insights into the biology of childhood cancer and structural birth defects, including the discovery of shared genetic pathways between these disorders. Since 2015, the Kids First program has funded whole genome sequencing for 20,000 patients and their families. The program's vision is to alleviate suffering from childhood cancer and structural birth defects by fostering collaborative research to uncover the etiology of these diseases and supporting data sharing within the pediatric research community. The Kids First Data Resource Center supports two tools for pediatric disease research. The Kids First Data Resource Portal allows users to query or search Kids First datasets, build virtual cohorts or collections of individual participants, and identify genomic files for analysis. Cavatica, built by Seven Bridges Genomics, allows users to analyze and visualize data using bioinformatic workflows in a cloud-based environment. Together, this powerful tandem of tools are accelerating research in pediatric cancer and structural birth defects. The Kids First portal includes several powerful features and tools that allow users to conduct their own pediatric research. The Studies tool provides a broad overview of the studies, participants, and types of data within the portal. The Variants tool allows users to search more deeply to learn whether specific variants of interest were identified in Kids First studies. This video will provide an introduction to two of the features of the Kids First portal: the Explore Data tool for building a virtual cohort of participants, and the File repository for identifying data files. From there, files can be analyzed in Kids First's cloud research platform, Cavatica, highlighting how an investigator could use each of these as part of their own research project. The Kids First Data Resource Portal allows users to query Kids First datasets, create their own collections of participants to study, and identify genomic files for analysis. Accessing the portal does require an account, but users can create a free account that enables them to easily view and browse data files available. To access Kids First genomic files for analysis, users require approval through NIH's dbCAP website. A user querying the portal typically begins with the Explore Data tool. This feature allows users to build virtual cohorts of participants based on specific search criteria. Beginning with the full dataset in the portal, users narrow the number of participants by applying clinical, demographic, or other filters. For example, a user studying the heart defect tetralogy of Fallot can identify participants with this phenotype by applying a filter from the Clinical tab. Notice how the active number of participants has decreased in the query. The user can apply multiple filters to the same query. For example, participants diagnosed with tetralogy of Fallot and abnormalities of the digestive system. Graphical visualizations update to represent the cohort within the query. These graphs can also be clicked on themselves to add more filters to the cohort. For example, selecting only participants that were diagnosed as newborns. The table view provides a listing of the participants in the active query. Each participant can be selected individually to bring up more clinical details and metadata. Queries can also be saved for further use or to share with collaborators. Once a user has finalized their query, the files associated with these participants can be browsed within the File repository. These are all the data files available for the participants within the virtual cohort generated in the Explore Data tool. Users can continue to narrow their file selection according to additional clinical or file filters. For example, a user looking to take advantage of the harmonized genomic variants produced by the Kids First DRC could apply a filter for these files in VCF format. Alternatively, a user looking to start their own analysis from the aligned reads themselves could apply a filter for just those in CRAM format. A file manifest, as well as a comprehensive clinical and biospecimen data, can be generated and downloaded for the files active in the repository. At this point, a user could download these files individually or analyze them in Cavatica, Kids First cloud-based analysis platform. The URL for the file repository updates with each new filter applied. Users can bookmark these pages in their internet browser or copy and paste them into an email to share with collaborators. Cavatica is a cloud-based analysis platform designed to handle large volumes of pediatric genomics data. It is produced in collaboration with 7 Bridges and based on the 7 Bridges platform for cloud storage and bioinformatics analysis. Projects are the core building blocks of Cavatica. Each project corresponds to a distinct scientific investigation and serves as a container and private workspace for its data, analysis workflows, and results. Multiple workflow executions can be carried out within a project. Access to a project is restricted to the collaborators in the investigation. Each project has at least one administrator who controls the project member's permissions. These permissions allow the project member as much or as little access to project resources as the project administrator wishes. Each project allows for bringing together data files that can be analyzed and visualized on the Cavatica platform. Files identified on the Kids First portal can be seamlessly pushed to a Cavatica project and appear under the Files tab, ready for analysis. A user can also bring their own data into Cavatica, either by dragging and dropping from their own computer, importing from an FTP server, or connecting their AWS or GCP volumes. Cavatica is more than just a data storage center, offering a repository of publicly available apps such as workflows and other tools suitable for many different types of data analysis. Publicly available apps are maintained by the Cavatica and Kids First Bioinformatics teams. You can search for an app by either entering the app name, a part of the app description, or the toolkit itself. Advanced users can also build their own apps and bring their own pipelines into the Cavatica environment. A Docker image containing the tool and its dependencies can be pushed to the Cavatica image registry, which can then be edited and transcribed automatically into the common workflow language. This means that there is no need to reconfigure your existing command line tools to meet any proprietary format. The same pipelines that the Kids First DRC uses to generate files available throughout the Kids First data resource are also available in Cavatica. Users looking to harmonize their own genomic data with Kids First data can upload to Cavatica and run these workflows to allow for comparisons across data sets. A task is an execution of an app. Each task is associated with a set of input files and parameter settings. Tasks can either be run a single time or in batches, executing the same workflow on multiple sets of files. After selecting the appropriate inputs, settings, and controls, the task is run using the Run button. Each tool that is run in a task is executed on a computation instance in the cloud. Instances are virtual computers. Different instance types have different allocations of CPU and memory, so are suited for workloads with different computational requirements. When a task is completed, it remains documented in Cavatica. The tasks listed on the task page are accompanied by their statuses, either failed, completed, or reported, and their initiation dates. To view more details about a task, including details of its outputs and task logs, click on the task's name. This allows for easy reproducibility of work in the future. Bioinformatics analyses are rarely complete with the execution of a workflow, and results often need to be further analyzed. The Data Cruncher is an interactive analysis tool on Cavatica for data exploration and visualization using familiar environments like JupyterLab and RStudio. When users need to run a few very simple shell commands to explore data or perform a more complex analysis using Python or R, the Data Cruncher will enable them to be able to do it directly on Cavatica, thus avoiding the time and resource-consuming process of downloading data to a local machine. The Kids First portal and Cavatica represent a powerful set of tools for research in pediatric cancer and structural birth defects. For more information, visit our website, kidsfirstdrc.org, or contact support@kidsfirstdrc.org with any questions.